This is my exam mini video for Genetics 311, Exam 4, and I chose Prompt 4, 1A. My name is Haley Honeycutt, and let's get started. So we're going to be talking about how a mutation that's caused by an insertion of one base pair can result in the production of a truncated, non-functional mutant protein. Truncated just means that it's short. So this is the coding dictionary that we're going to be using in reference to amino acids, or proteins you can call them, for the mRNA sequence. Given to us are the sequences of the normal and mutant proteins. Under each amino acid protein, I have listed the possible mRNA sequences for each amino acid protein. When comparing the two sequences, you can tell that the mutation occurred before the phenylalanine of the normal sequence. The mutation that occurred was an insertion of an A base. It is inserted in the normal sequence between tryptophan and phenylalanine. By looking at the mutant sequence, you can see that an A was inserted because isoleucine reads AUU, C or A. Now I'm going to show you how to figure out the rest of the mRNA sequences for both normal and mutant proteins. We know that both AUG and UGG are both given in the normal and the mutant strands. Now we have to make sure that all the rest of the bases match up. So if we insert an A in front of the phenylalanine of the normal sequence, that gives us an A in the mutant strand under isoleucine. We know that the next base has to be U because there's no other option in phenylalanine or isoleucine. The two U's under phenylalanine in the normal sequence match to the two U's under isoleucine in the mutant sequence. Next, we want to pick between the U and the C under phenylalanine, which we would pick the C because that matches the C under histidine. Both the A and the C under threonine matches the A and the C under histidine. Next, you have the option of picking U, C, A, or G under threonine. You would have to pick U because stop codons only start with U. Next, there are three stop codons to code from: UAA, UAG, and UGA. The two A's under lysine match with the two A's under stop codon. You cannot confirm anything else after the first two A's under lysine in the normal mRNA sequence. This is because the mutation resulted in the production of a truncated non-functional mutant protein. Therefore, all we know is that lysine can either be AAA or AAG, and glycine can either be GGU, GGC, GGA, or GGG. This was an example of a frame shift mutation because we had an insertion of one base pair within the gene that shifted the reading frame of all the codons following the mutation site. Here I've written our results for the normal and the mutation mRNA sequences. Both the normal and the mutant strand read from 5' to 3'. The mutation mRNA codes for AUG, UGG, AUU, CAC, UAA. The reading frame is shifted forward by one base. If you want to double check yourself, this is how you should do it. So looking back at what we originally had written down, we know that the AUG and the UGG codons match the mutation codons. Then you have your A, which is inserted between tryptophan and phenylalanine. Continue reading down the normal mRNA sequence. Start with your A, AUU. That's what goes under isoleucine. CAC is what goes under histidine. The last three that we can be sure of are UAA, which reads the stop codon for the mutant mRNA sequence. Everything matches up, and that's how you do it. Thanks for listening, and I hope you guys are doing well.